you. So I come from Vienna. I work in the group of Andrew Straw, and there we are trying to do what is written in the title. We are almost there, 50% reverse engineering that. But I'm not going to show you today any breakthrough scientific result. You can go and check the code for all this presentation in that Git repository there. So what is this about? Drosophila flies are a very good animal to study the brain. They are what biologists call a model organism that allows you to study how organisms are in general. It's complex enough that it's very interesting, but it's simple enough that it's tractable and you can study it. And you can see in those images a few of the characteristics. You can see the head of a brain, of a fly with the brain inside. You can see how big the eyes are and how they connect directly to the brain. And one of the most important aspects for the fly to be so useful as a tool for studying neuroscience is that it's very easy to manipulate genetically those flies. So you can target specific neurons and make them be impaired or activate them with lasers, etc. So you can study very easily the circuitry of the brain of a fly. So the other component we have in our group is virtual reality and tracking. So in the assays we just put animals into virtual reality arenas and we make them believe what we want them to believe. So in that way, we can just make them accomplish tasks or give them tasks to do. So not only we work with flies, we also work with fishes, with full spiders, and also with full GoPros. And we have Python in all those levels. So if you want to see some cool Python packages, you can go to the Straw Lab webpage and check in there. But today I'm not going to talk about the Python we use in those arenas for collecting the data, but instead I'm just going to talk about data analysis of the data that comes out those assays. So what we do is we have this large amount of virtual reality arenas, and each and every afternoon, night, a biologist comes there with a lot of flies, genetically modified or not. They put them in those places, and they make them just do exercises while exposed to stimuli. So with genetics and virtual reality, we have these assays in which for a night we just expose flies to what we want them to expose, and we make them fly. For example, in this case, we are forcing this fly to follow a loop just by using something called optomotor response. And by doing this with flies that are, for example, impaired and flies that are controlled, we get some nice data sets that are classification data sets. And then we can look for what differentiates trajectories of one kind of fly from another kind of fly. So this is behavioral sampling. Yeah?
Sorry. That bottom image is not virtual, that's a real fly flying.
That's a real fly flying. So the thing is, the animals are very small. You cannot see them in the videos. Okay? They're very, very small. But that thing that you see there is the fly, but it's that red dot while forced to do the loops. Okay? It's freely flying around. It just does whatever it wants, yeah?
Is this a product that's available for sale?
Again? Yeah, well, probably yes. So yeah, so we do. Yeah, you can control the mind of anybody, and actually we have many other mind control arenas in which we use, for example, lasers, and
so we're not
it's fine.
Here, we're just believing we're here, right?
Well, no, actually I'm here. That's the only real part of it. Yeah. Yeah. Okay. Well, I will continue. So, okay, so what do we have out of this? We have trials, okay? And trials are time series, loads of time series. So you can see there a top view of that flight that you just saw flying, and some time series associated with it. And so the amount, the quantity of data that we have in there is kind of large, okay? So a typical data set I was working with these days has over 300 experiments, which means 300 nights putting flies in. Of course, we have many, so there are like 10 experiments per night. Over 400,000 trials, which means 300 million observations. And all that accounts for 15 gigabytes in single precision worth of trajectories and time series to analyze, which might become very soon much larger. So we have these data sets, and we want the biologists to be able to analyze them. But usually these data sets are too big for the computers they use, and they are not really able to use clusters and all these kind of machines. And then they all the time ask me to analyze the data. So we want them to be able to analyze it in their computers. So we need, I'm going to talk now about three little libraries you can check out and that we use there, we have developed. The first one is a library we use for storing these trials. And well, how would you store these trials? So if you have gone to some PyData conferences, etc., you might have seen one of these talks from Francesc Altet or from Valentin Henel about compressed chunked storage. So these tendencies of today, we have these very nice libraries in Python that allows us to compress the data, retrieve it in chunks, retrieve it by columns, etc. And we have created our own library, which is called Jagged, and that allows us just to get this large amount of short time series fasters. There are lots of time series, they are short, so think about 400,000 pandas data frames of an average of 200, 300 number of rows. So this library has the simplest API that you can think of. So essentially, you just get one instance of one of those stores. In this case, it's a store using a library called BLOSK. You append just data frames, and you get them back. So you have append, get. And we have implemented loads of backends. Each of them has different qualities. Some of them are compressed. Some of them are chunked, columnar. They use memory maps. They allow you to retrieve any rows independently on which trial is without retrieving the whole trial. They may be lazy, meaning that you don't fetch the data when you read it. You can create even the pandas data frame, but the data is not yet in memory, so you can have 400,000 pandas data frames without having any memory there. And they allow you to choose which contiguity do you have. And there are lots of implementations, because we tried many things. So you can choose whatever you want. OK. The bottom line is that it works pretty well. So we were able to reduce this data set I was talking to you about from 15 to 11 gigabytes without suffering any delay on decompressing. More importantly, we can just retrieve any random selection of columns in very fast time. And yeah, the benchmark for this is in that repository. So the lessons learned by developing this library were that you really need to benchmark what really works. And in our case, our data is not compressible without preconditioning, which means first you need to shuffle it, so you put bytes that are similar together. This is provided by BLOSK. this library, which has these nice Python bindings. Our data pretty much likes, in many cases, like this. It's just a time series of slowly varying stuff. And so if you don't shuffle, you can apply any compression algorithm that you want, but you can't compress anything. If you shuffle, you do compress, and very much. Well, that depends a lot in the time series that you are looking at. So another lesson is that probably you should use different algorithms for different series. And yeah, I'm going to just go further. So how do we analyze these large collections of time series? Well, there are many ways, but we just go for the easiest, that is to extract features, that is scalars for each time series that tells about something about it. And we established this collaboration with these people from Imperial College. I was talking with them 2 days ago. And they have this huge library for extracting features from time series. It's called HCTSA, and it allows you just to tell many, many things about your time series, whether they are complex or not, things about their spectra, etc., etc. So we wanted to use this because we wanted also to collaborate with these people. But the library is MATLAB. So we actually paid a license for MATLAB that cost us €8,000 because even if we just look at flies, MathWorks considers us as a company. So we said, okay, what do we do? So we created this second library, which is called Pyopy, Python, octave Python, which is a library that allows us to talk to MATLAB and MATLAB people. Because these guys now really want to go to Python. I believe that we're going to switch there, maybe. And this is going to help to bridge it. This library is huge. It's a huge library. So Pyopy has these two goals. Helps creating Pythonic bindings to MATLAB libraries. This is done by parsing MATLAB code and just getting syntax trees and be able to say what is the name of the function, what are the parameters, etc. The second goal that has been provided by the first goal being achieved is providing bindings to this time series feature extraction library, HCTSA. So if you go to the web page, you can see a very big Python file that is called Python bindings. That is around 50,000 lines, and it's all populated of this kind of classes. Each class is one of the operations. And for example, you can compute permutation entropy, which is a feature which tells you some stuff about the complexity of the time series. And well, it's pure Python. When you use this, you use this kind of code. Just import the library, call prepare, which just spawns an instance of MATLAB, because this library does not translate MATLAB code. It just calls MATLAB. Just make it appear. Just say which things do you want to compute. For example, these two different versions of that feature I was telling about with different parameters. And then you just call it as if was Python, compute the thing. So it now really looks like Python, no going into MATLAB. OK, and the third library I'm going to talk about, it's called whatami, which is a library that works and is aimed at identifying your computation. So imagine you have these 200 functions, classes that compute different time series features, and they have different parameters. We can combine them in the way we want. How do you identify those things? OK, so this library works like this. Well, you import. It has several parts, but you import, for example, this decorator, and you just apply it to a class, a function, a variable, whatever you want. And then you get some functions like these ones, which allows you to get the identity of the objects just with a string and parse back that identity so you get the parameters. So imagine this is the superclass of all the feature structures, all our classes that do try to extract features from time series. That was enough. You don't need to do anything anymore for any other class. You get those identifiers just by extending the class. Okay, so this is an example on doing this. We get a feature structure that is this variable, which is just that feature structure with a particular configuration. And we can ask, what are you? And it returns this string. Okay. This string is just a Python call with all the parameters specified by name and sorted by key. So all the identifiers are always the same. And notice something. So we have this parameter here, return counts, but that does not appear in there. And the reason for that is that in whatami there are two types of parameters, those that change the result of the computation, those that don't change the result of the computation. Those that don't change the result of the computation are not part of the default identifier, which allows whatami to be used also for caching computations. And it's useful also, for example, to generate the feature names that are given by this computation. So here you see these 1, 2 features that are returned by that class when you pass it a time series. And we can manipulate those strings very easily. Okay, so this is a slide I'm just going to skip. But whatami is pretty cool, even if it sounds like it does a very simple thing. So just check it out. And I'm just going to finish with an example analysis in which I combine these 3 libraries. This is again the address of where the code lives. And it comes also with a data set of 1.7 gigabytes trajectories. So if you download it, you can just have the whole thing. So we give to our biologists nice representations, usually created with Seaborn. They love Seaborn. They love their documentation. And they're able to change parameters, make their stuff there. So we're going to compute. We're going to create this plot in which we see how the correlation of what the fly is doing with what the stimulus is trying the fly to do varies along time for different stimuli. So each and every of those 4 plots are correspond to different stimuli. Okay, so code review. We store the data somewhere. And we will work with this small data set I was talking to you about, which just has 52,000 trials inside with 80 million observations or rows. And it's worth 1.7 gigabytes uncompressed. You can download it, then you can just have a nice class that allows you to access all the different parts of the data. And then you can just put the data into one of those jagged storages. In this case, is a storage called jagged by matlab, which has this particular property that you can instantiate pandas data frames with the data from this storage, and they are just lazy, and they actually, you never go out of memory because this is mapped. So the operating system just evicts the pages when it needs to. Okay, continuing. We get a data frame which contains the metadata for all the trials, or those 50,000 trials, I think it was. Then, by using this partial, we can ask for a subset of the time series. So this partial is just over the get function of the jacked instance. And this is the type of queries we use. So we just get those trials that are larger or longer than 2 seconds for one concrete genotype. Here is where we get the time series. Okay. We define feature structures. Any list you want. So that's very easy to do. Compute the features. And then whatami allows us or help us to put, so we get a data frame of features. So each trial now is represented by a few numbers. And we can use pandas plus whatami to put that data frame into long form or tidy. I know probably you know this kind of speaking, which means that it's in a very standard way so that Seaborn very easily can ingest it and generate the plots. So to go into this long form data frame, this is a snippet of the function that does it. We use whatami to select from the feature names. The feature names are those whatami identifiers. So the provenance of the computations are still in there. And we use it to select just, if you see this, we are generating several kinds of features. For generating the plot I was showing to you, you only want these kind of features. So that's what this call does. Select columns based on what they are. And then we extract from those columns the lag, which is just one of the parameters. So here we vary the lag from -80 to 81, which means that we check how the fly is correlating with the stimulus at those times continuously. So now where the lag is a column in the data frame. And that's how you just plot it. You have a function to plot any kind of these melted data frames to generate these kind of plots that are very useful for biologists to explore the data. So that was all. And I want to acknowledge all the people that have provided me with the very nice images that you have seen. Thanks.
Thank you, Santiago. It was very interesting. We have time for a couple of questions. So, any question? No?
That's good.
Okay, then I ask the next speaker to connect the laptop. Thank you so much.
Thanks.